Welcome back to Nova Bio. Today we will talk about proteins, which is a very important chapter under biochemistry. So generally, proteins are what? They are building blocks of amino acids, and they play a very important role in biochemistry. So proteins are made up of polypeptides, and there are four structures under which proteins are classified, such as primary, secondary, tertiary, and quaternary. Now, we have learned a hypothesis earlier when we were small that one gene contributes one protein. Does that hold true? Doesn't, right? So, we have completed recently Human Genome Project, and from Human Genome Project we came to know that we are having somewhere around 29,000 approximately genes which are present in humans, okay, and they contribute for more than millions of proteins, right? So that was about overview of proteins. Now we will get back to the structure of proteins. Now proteins are divided into primary structure, secondary structure, tertiary structure, and quaternary. So we will see one by one. Now as you can see, primary structure, it is just a polypeptide chain, and it is having a end which starts with nitrogen and a end which ends with carbon. Okay, so it is just a polypeptide chain and it doesn't have any structure as such. But each amino acid or stretch of amino acid holds a code for a particular secondary structure. So that we will see in how we can predict a secondary structure based on primary structure or a sequence in later lectures. So in secondary structure of proteins, we have alpha helices and beta sheets. As you can see, alpha helices, these are ring-like structures. Now beta sheets, we have parallel beta sheets and anti-parallel beta sheets. You can see parallel and anti-parallel, right? So when alpha helices and beta sheets come together, they form a super secondary structure, which is also called a super secondary unit. So that is about a secondary structure. Now we have tertiary structures. Now the difference between secondary and tertiary structure: secondary structures are held together by intra-hydrogen bonds. That is very much important, intra-hydrogen bonds. Whereas tertiary structures hold together by van der Waals interaction, ionic interaction, salt bridges, disulfide bridges. So that is how tertiary structures made up of. Now if you look at this particular structure, can you point out a secondary structure present in that? Yes, you can. This region, right? So this is your alpha helix, and this region is your anti-parallel beta sheet, right? So this is how you can point out a secondary structure in your tertiary structure. So they are held together by many non-covalent interactions. Now quaternary structure, it is little bit complicated structure, and complexity of structures will keep on increasing from primary to quaternary. In quaternary structure, the best example which we have studied so far is hemoglobin, right? So hemoglobin, or you can say a myoglobin, they are the best example. They are made up of homodimers basically. So it contains two alpha chains and two beta chains which are attached to ion which is present at the structure center. So center of the structure is containing iron, and that is a prosthetic group, inorganic prosthetic group. So quaternary structures comes with a prosthetic group concept. So prosthetic group is a group which helps in function of a quaternary structure of a protein. If it is a organic, then it will be a vitamins. If it is a inorganic, it will be a metal ion. So they act together to give a function of a protein. So this is also made up of many non-covalent interactions such as ionic interactions, van der Waals forces, hydrogen bonds, salt bridges, disulfide bridges. Now the proteins which are present in cytosol doesn't have disulfide bridges because they are already present in reduced environment. Now this is a quaternary structure, and the example of that is also hemoglobin or myoglobin. So this was about proteins, and these were the four structure of proteins which we have studied. So this was about basic structural configuration of proteins. Proteins are very dynamic in nature. They keep on changing their structure. So if you have seen any protein in various conformations, then they are called as conformational isomers. About proteins, thank you for watching this lecture. Please let us know if you want any other particular lecture on such topics.